well, that coral's pretty characteristic, like that. And yeah, we see corals with that shape, but it will move around with the water.
So one of our viewers says that it was a mapping transit. Mike was on a cross Pacific from San Francisco, and we missed Mary on that cruise.
Well, anybody who's met Mike never forgets him. He's a character.
Yeah,
My buddy.
that's great. One very cool thing about being in the boat is that you get to meet very interesting people. And to be able to be here, I guess, like, we all share something in common. And that's something special.
I think I mentioned yesterday, I met a woman on the boat last year who lived in Latvia.
Oh,
And
yeah.
my sister and I went to Latvia this year, and I just call her up. I said, "Hey, Leva, we come by? Can we stop by and see you?" Sure, come on over.
So great.
I did that in Hungary this year. Met our old stewardess and her mother and her sister and her niece.
Where?
In Hungary?
Yeah, there's a lot of people who have come back, for several years in a row that I wouldn't mind calling up and saying, "Hey, I'm in this country. Can I come by and see you?"
Yeah,
It's really nice. And of course, any of the people from the boat I've met are welcome in Nashville. Come stay at my house. A couple people have come. A couple SCFs have been there.
I might be visiting all of you, so you better have
come
a
on.
little place for me.
I have a nice guest room. And a puppy.
Oh, I'm sold.
Of course, nobody knows anything about my puppy on the boat, but
Know its name.
What's his name?
I haven't shown everyone 14 pictures of
I
her.
absolutely believe that.
Wow, you can see all of the coral here, the color shining.
Depending on time, this might be a nice place to get a Niskin water sample.
Okay.
Yeah.
Sounds good.
Yeah, and then with that, we would want one of these small corals to kind of ground truth the results from the water analysis.
Yes, Becca, yes. Yes, yes, yes.
Which color coral would you like?
What haven't they got? Have they had one of the yellow ones?
They did get a yellow coral before, but I pulled up an image of it and it looks different from the
Okay.
ones that we're seeing here. So I think either one could be.
Okay, let's try for a yellow.
Okay.
Eeny, meeny, miny, mo.
The one, the lasers.
Yeah, I like that one. Good choice.
Waiting for the sunrise.
Oh, perfect. So, do either ROV have a magnetometer on board to measure Earth's seafloor magnetic field anomalies?
Well, we have magnetic-based compasses, but those we use to just measure our heading. It's not really looking at anomalies. Anomalies would probably just mess up our heading.
One of those magnetic compasses is inside a steel bottle.
Yeah, it works great.
Ew.
Sea cucumber. Yes.
Think I can get away with this before the ship leaves me?
Come down this way.
Ship is holding Argus
is swinging probably.
Yeah, we'll probably have another 10 meters. Still within tether.
We have a viewer that worked as a former JSON teacher trainer for the state of Alabama and worked with Bob when he visited Alabama.
They want just a little snip.
Do you know what they collected before, Becca? Was it just a snip or the whole?
Oh yeah, I believe it was just a snip of it. Yep.
Yep, let's just take a snip then.
You want me to go in?
Yeah, sure.
No.
Uh-oh.
Uh-oh. Those will work.
Now,
No one saw.
No.
that's really crumbly stuff. We could try and slurp that, I guess.
Yeah, I believe slurp jar number 7 is open.
Okay.
Swap
The
our
stuff that I...
cameras.
Pop them back.
You need a flush chamber.
Yeah, can you rig that around?
Yep. Should be flush. You wanna flush now?
Yeah.
Alright.
Do you guys have a, some of this is a bit branchy. Do you have a sample box you want to try and put it in, or just go for the flush?
We do have a small bio box open on the starboard side. It's the...
What I have in the manip now, I could put...
Yeah, let's try that.
Okay.
Because I think this will be our last sample, so.
Yeah, it's
Yeah.
in, it's D, so it's the aftmost.
Alright, forget
Do
the
you want-
slurp.
Do you want the starboard salvo setup or whatever?
Sure, yeah.
Yeah, very brittle.
Oh, that's not the right camera. Let's see.
So for our followers, part of the goals of the expedition is to better understand what
Oh, hold on. Keep that closed for a minute.
they
Get-
have on the
Get this
seafloor.
camera set up and follow the arm with the bubble.
Yeah, I didn't realize that one was swapped too.
And in order to do that, we need samples.
Yeah.
That's
Yeah,
why we're
just show me
collecting rocks,
The stall
Oh, that's
position for the
not-
bubble.
Bubble.
corals, and other living organisms
That's okay.
as well.
It's
Oh,
up
you want
there.
it up there?
Yeah, I don't care. I just need a
Okay.
little.
But we also make sure that
Just
we don't
up,
sample.
like the number 4
if there's only one.
Yeah.
of it.
preset would
so
be good.
This is
Yeah,
an area with
that's
a
what I
lot of
need.
corals, so we know that we're not taking the only individual.
So we're sampling different individuals of the same species, ideally, right?
Yes, yeah.
Mm-hmm.
And different species as well, so
Mhm.
something of interest.
Mm-hmm.
But I think we've been filling up Hercules for the last
Okay.
20-something
So
hours.
it's the furthest most
this will probably
small.
be our last sample.
Yes.
Yeah.
Want me to?
Yeah, go ahead.
So Georgia,
Yes.
to the left, that sponge that is covered with crinoids, if they keep inhabiting, like colonizing that sponge, would
Mhm.
that sponge die?
No, no, because those crinoids might move on and they're not, no, they're so small.
Okay.
Come in.
So if it was taken over by,
There's
you know,
still some.
zoanthid or
Yeah,
Yeah.
there's still a little piece.
some other encrusting,
Some of it definitely went in
then
there.
eventually.
Yeah,
but
it all
until
went in. There was only one
crinoids,
little bit was hanging
I don't
off
think
there.
that's a problem.
Alright,
It's all gone now.
we tray in.
All right.
We have a viewer asking if we could take what fell of the coral, or not really.
We could have. I think we got a fair bit in that bio box, didn't you, Mike?
Yep.
So I think that's probably enough.
Okay.
Becca, do you think that was enough?
Yep.
Yep.
Okay.
Yeah, so I think we have enough there to take with us, and then we'll just do a water sample as well in the same spot. So that coral will grow back.
So it's not that we destroyed it, right?
No, so we've left the holdfast is still intact, and there's still branches on that, so it will grow back. And as I said, there's plenty of those other
Doing a
individuals of
skim
the
now.
same species in the same area, so
Yeah, in the skin, yeah.
you know, we've
Yeah,
sampled
that's
this in the best
important.
possible way.
Yeah. That's interesting also. So sampling is not only just taking pieces of things. You actually plan
Hey, Mary,
the
can you
sample
get the
process,
iris
right?
open in there? We don't have the porch light anymore.
That's all open.
All right, it's perfect.
So we already fired 7 and 8.
Yep, I'm gonna go for number 9. Not gonna stay there, are ya?
Yep, I think you got it.
Yes.
Nice job. You make it look easy.
I do the same thing 1,000 times. Just
Don't be so modest.
It was a little bit of a fail with the coral. I didn't think that one would be that brittle.
Yeah, so brittle.
Yeah, that's definitely a hard coral. I think we have a genus, but I don't know if I can pronounce it. Perfect. I have a question for you, Mike.
Yeah. Shoot.
You know, if you go to the arcade and they have those claw machines where you can get the toys, are you good at that?
Yeah, they're pretty rigged, but probably I should give them a go anyway.
Yeah, you could win big time.
Yeah.
Probably a little different. A little less control there. Than
It would be nice to go to a fair with Mike.
Yeah.
Remember, those games were really hard, but some people were had them figured out. Some people would definitely.
Well, I think you'd have a head start.
Right, is that it?
Just a little bit of experience.
Yeah.
You'd have to close one of his eyes, though, to be spot on.
Let's blindfold him entirely. I saw that stretch and yawn, Mary.
Making more room for breakfast. Stretch.
I know it's steep, but is it possible to have a look at one of those pink coral?
Yeah, sure.
Thank you.
Okay, zoom in there.
Paragorgia?
Yeah, it could be.
So pretty.
Just having a look.
Seems to have stopped.
Yeah, just racking it in
heavy
and out,
coral
moving
ring.
it around a bit, eh?
Oh,
Still
it
there.
is.
Yeah.
Georgia, what
Yeah.
looks like wire entangled there, are those crinoids?
Nope, so they're brittle stars.
Oh,
Yeah.
okay.
There's a lot actually too.
Yeah, exactly.
Right, that's good, thank you.
All right, full wide.
So the assemblage has definitely changed now. It's getting steeper too.
Just to bring the ship in a bit. I'll zoom in there.
Yep.
Let me go in and focus, come back out. Oh.
Yeah.
Yeah. Okay. That's called ballpark focusing.
Hello, bro. Nice to have you with us.
All right, full wide. Out on the end of the tether. Not very good.
You're right.
Yeah, I think there's a few different species down there.
Mm-hmm. Yep. How deep are we now?
1,333.3 meters.
Perfect. Thank you, Mike.
It's a lot of threes.
A lot of threes.
Is that a sign?
Maybe.
Take that as you will.
There's a broken coral there.
So Georgia,
Yes.
we have a viewer asking about maybe the currents change since we haven't seen many manganese encrusted stocks here. But we are shallower now, right?
Yeah, so we started at 2,500 meters, we're now at 1,300. So, I mean, I'm sure the currents have changed. The ROV guys may have noticed the currents a lot because they're steering through them. Have you guys seen a shift in any currents?
Not really. There was a tiny bit of current kicked up out of the west earlier, but it was really minor.
Minor.
There's almost nothing here now. Sometimes I find on the seamounts and on the way down to American Samoa, maybe it was Jarvis or something. Sometimes just along the contours on the ridge lines, you'll get just really ripping current, just as it comes around like the feature,
Right.
and then you get over the rise and it's all gone.
Oh, there is a crustacean just to the right of the lasers.
Oh, yeah.
Well,
Can we take a zoom on that?
Hello.
yep, go ahead.
Do about 1 more hour of bottom time.
Okay. Oh, there's a... I was looking at a different one, but there is a crab on that coral.
Is this?
There is one just to
There's
the...
your other one.
Yeah.
I didn't even see that one.
No, that's good. Teamwork.
Let's see if I can go in on him a little bit.
Oh, look at it. So cute.
Beady eyes.
Yeah.
He's looking at us.
It's looking everywhere.
That camera's really vibrating. Okay, full wide.
Thank you.
Yep.
Yeah, that's what it looks like. It's vibrating from something that's on the Herc.
Yeah. Not sure if it likes having the other camera strapped to the top of it. There's a, that's there, and then there's a kind of a bungee holding on the back of the Zeus to take the, it's a lot of kind of Rube Goldberg machine on there.
It's not too bad though.
No.
Oh, yeah, yeah, yeah. You're right. So maybe the altitude might have more impact. Yep.
Go for video. No, I'm good. Thanks. Okay, bye.
So Miles, if we do an hour more of bottom time, how long would it take then to ascend?
About 85 minutes.
That's some quick maths.
I think
How-
you already did the math. Cheating.
That's why I said we only have an hour left.
How big do you think it was, that shrimp? Very little, right?
Yeah, I think it was just a little bit bigger than the two lasers, which was 10 centimeters.
Yeah, we should be clear here now. Zoom in there.
Yeah, let's have a look.
Go.
Yeah, go ahead.
Nice.
Oh, hello.
Is that a spider?
It's just no. Lobster crab.
Or a little crab?
And there's a lot of brittle stars too. Yeah, there's a little lobster.
And come out a little bit.
Yeah.
That's quite a large colony. Can you pop the lasers back on for me?
Yep.
So I can... Perfect, thank you.
All right. Full wide.
So what happens if we find a ship or plane that was not recorded out here?
I would have no idea what the protocol for that would be.
Was the little creature before a squat lobster?
It looked like it.
Yeah.
Yeah, and there was a lot of brittle stars on that colony as well.
Thanks for sending your jokes our way.
Well, you have to share the joke now.
Okay. So if a shrimp wanted to fight another one and said, "Are you looking at me?" Would the other shrimp say, "Well, duh. I have 360-degree vision, so I cannot not look at you." It's true, we all need breakfast.
On Halloween, I always ask the little kids, I tell them they have to tell me a joke.
Oh.
It sounds like some of the jokes I get on Halloween from the little kids.
Yeah, that's great. And to respond a little bit, the question about the wreck, if we found something like that, we would just mark it, but we wouldn't damage it, or we wouldn't conduct any sampling
There's a fish
nearby.
right
Yeah.
Fish?
there. Yeah, right in between the lasers.
Oh.
Let's zoom in a little bit.
Wow.
Oh.
He thought he could just hang there and we wouldn't notice.
It's gone.
Yeah, definitely not a lot of fish.
No. I wonder if that'll change as we move to different sites on this cruise.
Oh, are we actually in the sanctuary right now?
Yes.
Yes, we are. Okay.
Mm-hmm.
Yeah, we put swains at the start of the cruise because we weren't sure what the weather would do. So this is our furthest spot, so we were really hoping we could make it out here, and we did. And I think we're heading to a seamount
Yay.
next. There's that little channel there.
It's like a little watershed.
Yeah. It's very distinct too.
Mm-hmm.
It's very distinct sides.
So nice to have people from the Czech Republic, Barbados, Gibraltar, Spain. Thanks for following us. So if we mark any wreck like that, will we come back later or investigate or leave that to the local authorities?
Zoom in there.
Yeah, let's have a look.
Oh, that's nice.
He wanted us to zoom in.
Hi.
He came towards us.
Where is he? There he is.
Nice. Wow, look how... Oh,
Oh.
it's gone.
When they decide to go, they're gone.
Oh, they're quick.
Oh, you can probably
That's
get a
a
cool shot of this one. It'll be all dark behind it.
That's pretty.
That's beautiful.
Very, very pretty.
Hey, you want to zoom in there?
Yeah. Little squat lobster on the side.
Let me go all the way in and focus.
Yeah, sure.
And there's
is
a little
so
crab
cool.
there too.
Ah! Squat lobster.
Yeah, I think there's
Nice.
two.
Oh,
Yeah.
there are several.
That's
of
another big
those,
colony too.
Let's see if I can get the whole thing in there.
There we go. It's beautiful.
You're going like really tight there.
Focus.
Trying to get this. There.
Let me go in and focus on that one.
Yeah. I think I'm just up against a rock now. That's the end of my little track.
Your little pan.
Yeah.
It's nicely done.
All right. Yeah. It's all right. All right. Now
Great.
full wide.
Thank you.
The last cruise we found one of these really, not very helpful, I can't remember the name, but it was very, it's like a spirally curl, looks like or a coral looks like I don't know, it winds up being tight and round, but if you get in and look at it sideways, it's like a coiled spring almost.
Oh.
And it was out on a ledge, kind of like better than that, so you could get behind it and just have like that thing lit up really well with just black behind it.
Yeah, that was a nice shot.
Do you think that track was the result of shifting plates?
I wouldn't like to say. I'm not sure. Some things definitely cause that. It's very distinct boundaries on that. So some sort of change in geology.
Want to zoom in on that?
Yes, please.
Go ahead.
It's a very nice anemone.
So cool.
Be cool to see that little mouth open and suck something in. That'd be neat.
Okay.
So Georgia, we have a question about
steep here again.
why corals retract their
Which
polyps.
should be pretty stable.
Is it because they are full up with food, or are they sleeping, or what would be the cause?
So there's a variety of different reasons that they would retract their polyps. So one would be it's a protection mechanism. So up in the shallows, a lot of corals feed at nighttime because there's less fish active. So they keep their polyps, everything retracted during the day to protect themselves from hungry fish. And then they will extend them at night to feed. So down here, that could come into play as well. Also depending on current too, so some of them won't want their polyps extended, the tentacles extended if there was a very, very strong current. Others will. So that will come into play as well.
Want to zoom in there a little bit?
Yeah, let's take a look.
Just fly around it for a minute.
Yeah, so some will be feeding. So here's a good example. Some are out and some are in. So some of those are actively feeding and others are not. But anything that they catch is shared. So they all share.
Would they be feeding 24/7, just alternating different polyps?
Some might be.
Okay.
Thank you.
Come wide.
So Mari, we have a viewer that's some of us are old but still useful, and this viewer remembered Jason dives with Alvin and when Bob found the black smokers. And also the viewer says that he or she grew up watching Jacques Cousteau, one of the men who inspired your generation to go explore. And actually not only your generation. I grew up watching Jacques Cousteau because my dad like pushed me to do it. And I got super inspired by that. I am here because of him, basically. So,
That's cool.
yeah.
There we go.
I grew up in the desert. I didn't know anything about the ocean.
I grew up in Ecuador in the middle of the mountains. So we're like...
At least you had water.
Not very close, though. But yeah, different from the desert, for sure. How did you get involved with the ocean then?
I
Like-
just accidentally tripped into it when I was working on Jason project.
Cool. We have an interesting question about the biology at this depth. Do they know the difference between night and day?
No, because
That was it, no.
there's no change here in light. Yeah, so that comes in to play a lot more in the shallows.
So how would they have cycles then? It's not day-night, it's just about the availability of nutrients, or?
Yeah, and a lot of it's chemical cues. So corals anyway tend to work on chemical cues. So corals will spawn. Most hard corals, this is, spawn in a synchronized way. So within a few hours of each other to maximize their reproductive efficiency. So a lot of them, have worked on chemical cues. So yeah, there's a few differences with the deep sea corals. But it looks very different up here.
Yeah, a lot of change.
we still at 1,300 meters depth, more or less?
Just about, yeah.
Okay. Yeah, very different species of corals here than compared to the surface or the shallower waters. And yeah, that's an interesting thought about deep sea species might have tidal or lunar cycles. Someone has to do with those cycles, maybe, yeah. And for the people that just started following us, we are exploring the National Marine Sanctuary of American Samoa. And this expedition will started on July 23rd and will continue until August the 5th. And the idea is to build upon the data collected in 2017 by NOAA ship Okeanos Explorer. So we will acquire some baseline information on deep sea and mesophotic habitat in American Samoa, taking some samples, geology and biological samples. Yeah. And also for sure searching for potentially new species of deep sea corals. And right now we're close to Swains Island, the northern part of American Samoa. Ave, are you still with us?
Can you still hear me?
No. Now?
Speak louder.
Oh, yeah, I'm still here.
Yeah, we have some people from American Samoa now. So would you like to share with them what is the most amazing thing you've seen so far, or the most, I don't know, the thing that you've liked the most?
Okay. E whatalo whatu nisi fo e ro mai mō memory ka Samoa o mātolo o whamautua ni East Wayne. I le ngā whewtau tantanga ai mai te le lawani amu o loo iei I le I vai ngā sami wheo o Swain I a mautou silofia o Swain o se sifo I lea paka whāsao Amerika Samoa. Ia a whai e into vesili ia vesili mai nisi o whai a onga po saintisi fo I olo iai nei. Ia whamaro lava whaftai le tantanga I mai I le nei fo ia susuenga whaftai. Thank you.
Malalava.
Thank you, Abby.
You're welcome.
Yeah, I think American Samoa is just getting up. Ready
Yeah,
to start
just getting up.
their work day.
Yeah. Thanks for being with us so early in the morning.
Yeah, I'm feeling a zoom, if possible.
Yeah, go ahead. Yeah, go in. Yep.
Ah, so cool.
Very nice, thank you. Yeah, it's hard to focus when when they're moving.
So the shallower we go, the more fish we would be
Want me
able
to
to
come out?
find, right?
In theory,
In theory.
but this hasn't been a particularly fishy site. So,
Yeah, not really.
we've seen a few, but not very many. So maybe as we get a little bit shallower, but we're also running out of bottom time now. So it'll be interesting to see the other sites that we go to, whether there's more fish or not. But as I said on a previous watch, it could be that
come out.
there are fish around, but they've been scared off by the noise of the ROV. So
We are taking samples of the water here, right?
we've taken a few, yes. So when we sampled that coral earlier, we took a water sample as well.
So we have a question about what is the acidity of this water compared to the surface. Maybe after processing those samples, we'll know more about that, or do you guys know something?
I don't think pH is something that's actively measured during the dive, but it might be possible to figure that out.
After?
Yeah, after.
And do we know if the water temperature is rising equally to the surface temperature at this depth? That's a good question.
So far, it looks like it's been pretty constant so far in the dive, but maybe as we get into an area where the sunlight can penetrate the water, we'll see an increase in temperature.
And also it's interesting because we don't have much information about this area from previous decades to compare to see.
Right. So a lot of the information we're collecting now is baseline.
Exactly.
So it's our first set.
That will be difficult to compare in terms of like rising temperatures or climate change, right?
Right. So what temperature are we at now?
Looks like 3.5.
Little chilly.
Is that in
Celsius.
Celsius? Okay. Yeah, that's chilly.
We
I
won't
would
see
like to swim there.
a big temperature change until maybe 700 or 800 meters. And then it'll increase really rapidly.
Water is like very turbid all of a sudden.
Yeah, it is.
Suddenly it got murkier.
Looks like it was a nice sunrise this morning. You can see on that camera. There's not much down there. It's changed.